Welcome. My name is Dave Arnold. I'm the Executive Director of NextGen Precision Health Initiative. We're here in Kansas City at UMKC. Before I start the introduction, I do want to make one announcement. So because of space restrictions here in Kansas City today, we have to have a 5-minute intermission where we're going to move from one room to the next. And so the question and answer part of today's seminar will be after a brief pause. And so there'll be people outside the door here to guide you to the room. So you're going to go down the hall, 3 floors, and there'll be banners showing you where to go for the question and answers part of the session. So it is a great privilege to welcome Dr. Simon Friedman today to give our talk. Dr. Friedman is a Curator's Distinguished Professor in Pharmacy at UMKC, and his work is at the intersection between biology and chemistry. And today we're going to hear about how we can use light to control biology and to control the delivery of therapeutics. And so without further delay, welcome.
Great. Thanks very much. That's an encouraging start. Thank you very much. Okay, thank you for that introduction and for the invitation to talk to you today about some of the work that my lab is doing using light to control biology and the delivery of therapeutics. And this of course, fits into this umbrella of precision medicine. And when you think about, or when I thought about precision medicine, it reminds me of my college roommate who is from Sweden, and he enjoyed drinking milk, large quantities of milk, and had no problem digesting it. Whereas if I drank even a fraction of that, I would get an upset stomach. So, and now that difference might be due to genetics, it could be due to epigenetics, it could be due to microbiome issues, it could just be due to environmental differences. But the bottom line is, if something as innocuous as milk can have such a different reaction with individuals, what hope does medicine have? We know that medicines work. If they didn't, then the FDA wouldn't approve them. But working just means that some average dose given to an average patient will have some average positive benefit. But baked into all those averages is a lot of variability, ranging from complete inactivity in some individuals to toxicities in others. So there's a real need for better control of therapeutics to better match what they do with individuals whose needs can really vary all over the map. So, a need for control, but how? Well, we've been using the tool of light. And why light? Well, because light itself is very easy to control. It's easy to aim light where you want it. It's easy to determine when the light goes on. It's easy to determine how much light is being applied. So once you've brought a process under the control of light, you can control its spacing, its timing, and the degree to which it happens. So, in the process of doing that, we are chemists, and so we use the tools of chemistry because they're sort of uniquely powerful ways of interacting with biological systems. And I always have this observation when I know people who are chemists, and the route that they took to get there is, as children, there's sort of the blowing stuff up route, and the second route is the building things out of Legos route. And I was maybe a hybrid. I was maybe 30% the blowing stuff up, 70% the Lego route. And the Lego route is, it's actually a pretty good metaphor, I would say. It's, well, not a bad metaphor, because in chemistry, we start from a small number of building blocks, individual atoms, but more likely individual fragments that you can then link in just infinite number of ways. I mean, literally infinite number of possibilities for the way that we combine atoms to make molecules with interesting and useful properties. So, I'm going to tell 2 stories that fit into this overall idea of using light. And the first one is a more sort of fundamental biological story. It's about using light to control gene expression, which is really the foundation of what we're doing more recently, which is using light to control drug activity. But I wanted to tell both of these stories because they're really linked and they have some common themes that are kind of interesting. So, the spacing, timing, and amount of gene expression is critical for all of biology. And when I talk about gene expression, genes are these segments of DNA that tell the cell how to make proteins, and proteins are really the machines of biology. So as you're eating your food, there are specific proteins that are degrading individual proteins or fats into constituent pieces. As you're seeing me, there's a protein in your eye that's converting light into an electrical signal that the brain can interpret. As you're moving, there's a protein in your muscles that's converting chemical energy into motion. So all those proteins are coded for by genes, and the amount of those genes that is expressed and where they're expressed and the timing of that expression is absolutely critical for essentially all of biology. And this is particularly true of developmental biology, where when you start with a fertilized egg, it divides and you get sort of an amorphous blob. And at some point, this amorphous collection of cells has to figure out which end is the head, which end is the tail, which is the left side, which is the right side. It does that by having differential expression of proteins and gradients across the group of cells. So we thought it'd be really potentially very useful if we could control the spacing, timing, and amount of gene expression using light to essentially make a tool that developmental biologists and biologists in general could use to pick apart the role of gene expression in all of biology. So how to do that? Well, we turned to small interfering RNAs, which were, or still are, a powerful tool for reducing any target gene's expression. And this is one depiction of an siRNA. It's about 20 bases of duplex RNA, and it can target any gene based on its sequence. In this case, we target GFP, green fluorescent protein, because it's a really convenient readout. It's green, it fluoresces, it makes light, so it's easy to assay its presence. You just use a microscope or a microplate reader. And so a cell that expresses GFP will be green and fluorescing, but if you put in an siRNA that knocks down its expression, it will lose that fluorescence. So this is a model system that we use to understand the process of gene expression. And we asked the question, could we control those siRNAs using light? So how to actually do that? Well, after a lot of, each of these slides is distilling a lot of man-years of effort, but we came up with this approach where we attach to the ends of these RNAs blocking groups, which actually prevent them from having their activity. And they're light cleaved, which means when you apply light, they come off. So in the absence of light, those groups remain on, and cells continue to express GFP. In the presence of light, those groups come off, and you get active siRNA, which is able to reduce the expression of GFP. So yeah, every picture you can easily just draw it, but then how do you actually do it? So how to make these blocked siRNAs? There are a few different ways of depicting the siRNA. I've started with the most low resolution version, just showing the two strands, but that covers up a lot of complexity. There's about 1,700 to 1,800 atoms in one of these pieces of RNA. And doing chemistry on something like that can be very challenging because there's a lot of potential sites of reactivity. But what we figured out is a way of targeting just basically 4 groups out of this mess of atoms for specific reaction. In fact, it's these terminal phosphates that are present or can be present in the siRNA. And I'll talk a bit more about that momentarily. So this is our magic molecule that allowed us to make those modifications on the termini of the siRNA to introduce light cleavability. And it has multiple functions. These are sort of the bricks that have been connected to each other, the Lego bricks. On one end, we've got this diazo group, which turns out to be highly specifically reactive for those terminal phosphates, not for any of the internal phosphates, which was quite an interesting fortuitous discovery. On the other hand, we have this large, bulky group, which once it's attached to the siRNA, we think blocks its interaction with the cellular machinery that allows it to have activity. And then in between, we have this orthonitro group, which allows for light cleavability. So it's sort of a trifunctional molecule, which allows us then to do a very simple reaction to make our final protected siRNA. We simply mix those two things together. Because of what's called regiospecificity of that diazo for these terminal phosphates, we get a clean single species. And chemists, we all love clean single species. It's just very appealing to us. We don't like heterogeneous mixtures, and that's one very nice thing about this kind of chemistry. We make a single species with 4 modifications. And how do we know we've actually done that? Well, we've been very fortunate to have a pretty high-end mass spec in our school, and that was really pioneered by Bill Gutile and with the support of our administration over the years, where we actually can confirm that we have this tetra-modified siRNA. And with that in place, we can also demonstrate, and I'll have some actual data here, not just pictures in a moment, that when we irradiate this, we get native active siRNA being formed. Those groups come off, leaving behind no trace of their former presence. And so with that in place, we can now create patterns of expression with light, which was the whole aim to begin with. And how do we do that? Well, we're benefited from fabricating in-house small point sources of light, so light radiating from a very compact point. And because of that, the rays expand outwards, and if you have a mask in the way, you get a very sharp pattern of light then falling on the cells of interest. Now, this is kind of similar approach that's used for making integrated circuits. You have a light source, a very focused light source, and then a mask, and then a pattern being created. And what happens then is, in the places where light lands, you get this uncovering of native siRNA and hence knockdown in those regions. In regions where the light is blocked by the mask, you don't knock off those groups and you don't get reduction in GFP expression. And therefore, you'd expect to see some glowing cells in the pattern of the mask. So we can now create those patterns. And we did a lot of different patterns. One of the more eye-catching one is making an alphabet of letters. This is a little worried about that. But hopefully you can kind of see each of those letters is maybe a couple of millimeters across and is made up of a few hundred GFP expressing cells. We have them in both positive and negative, and the red is a type of control. So we're able to make very detailed patterns of expression using this approach. And furthermore, we wanted to see, could we make gradients of expression, because that's particularly useful for developmental biology. And in fact, so to do that, you have a mask that itself has a gradient where it's completely blocked at one end, completely transparent at the other end, and linearly varying in between. And indeed, we get this gradient of expression across the well. And again, this is a few millimeters across. So we were interested, once we had that in place, we wanted to see, could we do some fun biology with it? And we turned to the patterning of cell adherence. It turns out there's a gene called P10 that if you knock down its expression, the cells that have it, that expression knocked down, will unadhere from their culture surfaces. And so there's an interest in tissue engineering of making patterns of cells, but typically this is done by patterning the surface, the chemistry of the surface that they're being cultured on. We wanted to see, could we pattern them by patterning the inherent biology of the cells. And so we spatially targeted using an siRNA that was targeting a light-activated siRNA that was targeting P10. And we have full light, complete block, and then a pattern. And in fact, we get that pattern of cells. Now, this is not, oh boy, yeah. Oh well, there's a pattern, hopefully you can get the basic idea. The pattern of the mask is, yeah, that'd be great. Yeah, that's a bit better, thank you. That recapitulates the pattern of the mask. Now these are not GFP expressing. This is another dye that's got the same color, but is just showing the presence of the cells. It's a general fluorescent stain for the cells. Okay, so that we published a lot in that area. And it was very productive. And we learned a lot from doing that project, independent of the specifics. We also learned some broad themes. And those are that we can do chemistry on macromolecules if we figure out the chemistry well enough to make it specific so we get clean products. Things like RNA, which have thousands of molecular weight, we can do chemistry on those. We can use mass spectrometry to figure out what the products are and to really characterize them. And then we can control the activity of those large molecules with light. So those were the lessons. And at the same time, those lessons really suggested another potentially useful application, and that's kind of what we started off with, which is the controlled release of drugs. And really an ideal application of that is insulin, because if there's ever been a drug that needs variable delivery, it's insulin. So as you eat a carbohydrate-rich meal, it gets into your intestines, it gets broken down, the starch gets broken down into individual glucose molecules. Those get absorbed into the bloodstream. And if you have a functioning pancreas, the pancreas senses the presence of those of that glucose and releases insulin, which is then the signal to the body, to the liver, and the cells of the periphery to absorb glucose, which can then be used as an energy source or as a building block. And that's great. That's part of physiology. If you're type 1 diabetic, however, your beta cells no longer can make insulin, insulin isn't released, and glucose isn't absorbed from the bloodstream. And it turns out, I didn't quite appreciate it at the time when we started this project, but glucose is quite a toxic molecule. It sort of surprised me. It has this aldehyde group, which is very reactive towards amines, especially protein amines. You get modification products, ultimately cross-links, advanced glycation end products, which all lead to microvascular damage and a whole host of sequelae that really descend from this very simple reaction of the glucose molecule with the amines that are present on the proteins that are ubiquitous in the body. So there's a real problem with in type 1 diabetes because you get massive, a wide range of problems linked to those modifications, including neuropathy, and other microvasculature damage. But fortunately, about 100 years ago, just over 100 years ago, insulin was discovered. And this is really a triumph of let's call it modern biomedicine, because shortly after it was discovered, it was isolated and then made in large enough quantities that it could be given exogenously to people who could not make it themselves. And it really has saved millions of lives in the intervening 100 years. So insulin is fantastic. It's, as I said, it's saved many lives, but it's very tricky to use. First of all, it's a protein, so it can't be orally active. It would get digested as soon as, in the same way that your sandwiches would be. So it has to be injected multiple times a day, 2 to 4 times a day. You don't need much of it. It's a very small amount, but you need it in highly varying amounts depending on what you're eating, the volume, the nature of the calories, your general activity level, your inherent physiology, huge amount of variability of what you actually need. And it doesn't just vary between individuals, it varies continuously, temporally throughout the day. And because of that, diabetics are really burdened with multiple injections per day, every day for the rest of their life. And because of that, because they're doing individual injections, there's always a danger, in fact, a likelihood that you're going to be a mismatch between what you're administering and what you actually need. It's going to be over or under, because it's a step function versus what you really need, which is continuous variation, which is what the healthy pancreas does. So the challenge of the field after the discovery of insulin and the benefits that it provided, the challenge for the field then was to figure out a way of minimizing injections to get rid of that burden, and then to better match what is being delivered to the individual patient with what is actually needed to better match the actual blood glucose. This is a solution, the artificial pancreas, and this is the first generation of this kind of thing. It's kind of an Apollo era version, and this has become much more streamlined, but it essentially has these 3 components. It has a CGM, which means a continuous glucose monitor, something that measures blood sugar continuously throughout the day. It's got an algorithm that takes that information and makes a decision how much insulin is needed. And then it has a pump, something that delivers the insulin from outside of the body into the body. Now that contraption that was on the previous slide has gotten more streamlined over the years. It's been markedly improved. CGMs are much more compact. There's a lot of work proceeding to make them completely non-invasive using just optical signals. Algorithms have gotten better. They use machine learning and artificial intelligence to adapt to what the patient is experiencing. But pumps are basically what pumps have always been. A reservoir outside of the body, a mechanical device that then drives the insulin through a cannula or a microneedle across the skin into the body. And that physical connection, which is really inherent in insulin pumps, is really linked to a lot of different problems. And this is from an ad from a pump manufacturer, and my standard joke is every one of these ads shows some sort of sexy stomach, but there's nothing sexy about an insulin pump. Lots of problems associated with them. The first issue is that you get crimping and snagging, which can occlude flow. It's not really conducive to an active life. Second is your skin is continually breached, so there's a significant potential for infection. Because you try to make the orifice of these cannula as small as possible, you get biofouling because insulin has a propensity for aggregation. So you get biofouling and complete occlusion at times, leading to misdelivery and hyperglycemic excursions, and then thickening and scarring of the tissue at the site of injection. So there are a lot of physical problems associated, or problems with pumps that are associated with that physical connection. So we aren't the only people to have made this observation. People have called that infusion set, which is the link to the pump, as the Achilles heel of continuous insulin infusion. So some years ago, kind of emboldened by our success with the control of RNA with light and building up a chemical tool set to allow that to happen, we thought, is it possible to control insulin release using a photoactivated depot, using light to control its release? We call it the pad or photoactivated depot. And this is the overall idea of it. We have a material here shown in this green ellipse that's injected into the skin in the standard way that you would with regular insulin. But it's at the site of injection, it's insoluble. It stays at that site of injection, and it's inactive. It doesn't release insulin until it's stimulated by a light source through the skin, and then those blue dots, which represent insulin, are then released. And the idea is, if the chemistry is right, then more light will mean more insulin is released, less light means less insulin is released, and so you get that continuous variability you need from a pump without the physical connection associated with it. And furthermore, the idea is to cover that site of injection with a light source, maybe the size of a watch or even smaller, that both shadows the site and also gets information from a CGM, from a continuous glucose monitor, to give pulses of LED light. And the idea is each pulse of LED light will give a pulse of insulin that's released. And with that combination of continuous glucose information, as well as continuously variable release through these pulses of LED light, you can get good control, or better control, without the invasiveness associated with a pump. So how to actually do this? So we've got that the ellipse representing that material. So what does it actually look like? And we're going to bore in on the atomic details. The first generation material looked like this. The green wavy line is a polymer. We call it an insoluble matrix. The purpose of that is really so that when you inject the material, it stays at the site of injection so you know where to send the light. That's a critical piece. It's connected to the blue dots of insulin by these red squares, which are light cleave linkers. These are groups that, when they absorb the right wavelength of light, will break their bond with the insulin and allow it to diffuse away from the depot, be absorbed into the microvasculature, and get into systemic circulation. So that's one level of abstraction down. So how to actually turn that into a real material, how to actually construct this. Well, we started with insulin, and it's a protein, so it's somewhat complicated, but it's still amenable to chemistry. That was our feeling based on our work with RNA, that we could potentially do chemistry to this. And I won't get into all the gory details, but just this bottom molecule is our magic molecule, well, our first magic molecule. We've had a few magic molecules along the way. But this bottom molecule is the key one for this first generation material, and it has multiple pieces as well. It's again got this diazo group that turns out to be quite specific for the carboxyl groups on insulin for reaction, to link it to insulin. And then once it's linked, at the other end we have this azide group, this N3, which will allow us to connect the whole modified insulin to the polymer ultimately. And in between all those is this orthonitro group, which confers light cleavability, which is the ultimate thing we want, or to allow photolysis. So when we mix these two together, we can separate them, the species on an HPLC. We get 3 peaks, which indicates 3 products. And so with the power of mass spec, which again comes in very handy, we can tell that that first peak is unmodified insulin, so it didn't react. The next peak is singly modified, and the second peak is doubly modified. Now we can use all these species, but for this particular project, we used the singly modified. And we could then take that material and then do the famous click reaction of it onto a polymer. So the resin is shown in green here, and the final species is at the bottom. We have the green resin, which is this thing that confers insolubility. We've got the insulin on the other side, and then we've got this light cleave linker in between. We do all kinds of chemical characterization. I'm not going to dive into that, but the most sort of, the minimum thing that we need to see is that when we irradiate this with the right wavelength of light, what do we get coming off? We get a species, 5808, which is the mass of regular human insulin. So at the minimum, it's consistent with insulin. It doesn't mean it's bioactive necessarily, but it is at least, it hasn't lost atoms or gained atoms. So it's a good starting point. If we take that material now, which is a polymer with this insulin grafted onto it, and put it in a tube and administer pulses of light, we can look at any insulin that's been released into the supernatant, into the fluid above the resin, which is similar to what we'd want to actually do in a clinical setting. And so this shows that process. We're alternating periods of light and dark, pulses of light, periods of dark. And with each pulse of LED light, we get a pulse of insulin being released into the supernatant. In periods of dark, there's none released. Longer pulses have more insulin being released. Shorter pulses have less insulin being released. So this is about as good as we could have wanted from a first generation material, in that we could actually toggle and meter out the amount of insulin by varying the amount of light being applied. So we published that about 10 years ago at this point. And so that's for us, that was exciting. It was an in vitro success, but there are many ways in which this could fail in vivo. And my study section was happy to enumerate all of them. But eventually, see, we wanted the funding to figure out if it would work in vivo. They wanted us to tell it if it would work in vivo before they would give us the funding. So we sorted it out eventually. We came to an understanding. So we needed to see if it would work in vivo, because there are many ways this could fail. Not enough light could get across the skin. We're not transparent. The insulin could have lost its bioactivity, and so forth. So we turned to an animal model of diabetes, a rat model, and we did all this work and continue to with Dr. Karen Cover, who's at Children's Mercy, and so we're very grateful for that interaction. And so these are animals that do not make insulin, and so are hypoglycemic. And we also have to make a small light source that is light enough and small enough that it can be used on a diabetic animal. So we do all our fabrication in-house. This is about the size of a quarter, but we can make them even smaller than this. And there's a few tricks where we use magnets. There's a base plate with a hole in it, so we can put that on top of the depot, injected depot, so we know where it is, and then the light source can click on top of that with the magnets so that the light is registered with the injection site. So we know where we're sending the light, which is a good thing. And then we can control that with a control box. And this is what it looks like with an actual diabetic animal. We have the depot injection site into the skin. Then we have the base plate put in place, so with the circle circling where the injection is, and then we click the light source on top of that, and it's ready for irradiation. So we have both experimental animals and control animals. It's, we're scientists, so it's always good to have controls. Our controls and experimental animals both have the depot injected. They're both irradiated, but the control animals, the light is blocked with a sheet of aluminum foil. The idea being we capture any of the warming that might take place from the light source, so we eliminate that and so really the only difference between the experimental and control is the light hitting the skin. And this is our first in vivo result, and this is showing blood insulin versus time. The gold bar is the period of time of irradiation. Red points are control animals, blue points are experimental animals. So first observation, no release from the control animals, no leaching, which is great. You don't want to release insulin in the absence of light. We get a nice spike of insulin coming off and then a gradual decline, which is pretty typical of insulin. It gets cleared from the bloodstream by existing mechanisms. And then something we didn't anticipate, which is that we saw insulin in the bloodstream in 5 minutes. And we didn't really appreciate it at the time. I had to have enough clinicians sort of hit me over the head with the point that seeing insulin in the blood in 5 minutes is a really good thing therapeutically, because it means that you can quickly address rapidly rising blood glucose levels, which is very difficult to do. In fact, the normal insulin has maybe after injection you start getting observation in about 20 minutes. So you have ultra-fast insulins, but this is as fast or faster than all the commercially available ultra-fast insulins. And it's accomplished with a native insulin. This is regular human insulin. It's not a mutant insulin, which might have advantages. But the very minimum, having it be this fast turns out to be useful. There's so many things that go wrong in science that you don't anticipate. Every now and then, something like this happens where it's an unexpected real boon, and that was it. Now, this peak we're getting is actually not, it's kind of sub-therapeutic. It's not as robust as we'd like, but we'll talk about how to address that in a second. And so in this same experiment, we also looked at blood glucose, because you could have insulin that is released, but have it not have bioactivity because of all the chemical processing and so forth. But we looked at blood glucose and experimental animals. We did see this reduction, but it was not particularly robust. That was consistent with the lower amount of release of the insulin. So we asked the question, going back to that original data shown here, we have that initial irradiation, and you get this spike coming off, and then around the 1 hour point, it starts to decline. We asked the question, well, if we came in with a second pulse of light, would we get a second pulse of insulin and then a more robust drop in blood glucose at that point? And so we did that experiment, and this is a naive set of animals, a separate experiment, and with irradiation, we get that initial pulse of insulin and then a decline, and then at 60 minutes, we come in with a second pulse of light and we get this second pulse of insulin, which was very reassuring to us. It meant we could actually, in a living diabetic animal, we could meter out insulin by metering out the light. And with this in place, our blood glucose, this is now blood glucose versus time, we got this nice substantial 45% reduction in blood glucose. So both we were able to achieve a more therapeutically relevant level of glucose reduction, but also we showed that we could actually meter the insulin with the light. So, that was published, about 6 or 7 years ago at this point. So, and that was our first generation material, in vitro and in vivo results. And that material is fine. It was fantastic for beginning to understand how this process could potentially work. But it had, like a lot of prototypes, it doesn't look anything like the final thing, not that we're at the final thing yet, but it has problems associated with it. The first thing is it's very low in density. That's because there's a lot of polymer, which you need to make it insoluble, and not much insulin. And for the purposes of just proof of principle, that doesn't really matter. But in terms of practical long-term consequences, it is a problem because you really want, higher density means the depot can last for longer periods of time. So if you want a full day of use or potentially a week or even 2 weeks worth of use, higher density would be much better. These first generation materials are about 5% insulin, 95% other stuff. The other thing that higher density gives you is greater ease of photolysis because the photocleavable groups are in greater density. That makes them easier to impinge with the light rays that are coming through the skin. So it makes it easier to release the insulin. So density was a major aim. The second problem with the first generation material is we had this polymer that we were using, but it wasn't biodegradable. That was the long-term interest. But when you start actually thinking about that as opposed to just putting it on a slide, you realize that that is a major design challenge in and of itself because you have to make a material that is stable during the lifetime of the depot, but then biodegrades after it's done. And that is doable, but not trivial. So we're interested in capturing all the success of that first generation and getting rid of this density issue and the polymer, maybe potentially completely. So we thought about what are the actual key elements that we need from a material, because that's useful in figuring out designs. You can figure out what can then meet those requirements. And the key requirement is that it's insoluble prior to irradiation. Why? Because you want to inject a material that stays at the site of injection so you know where to send the light or place the light source. And the other thing is that after irradiation, you need to release soluble, native, bioactive insulin. If you have those two things, you have what you need for the system. So over the years, we've come up with multiple possible solutions. And I always say, sometimes you have an individual slide that literally represents man-years. I'm going to have one slide here that actually has 3 separate projects, each of which represents that, and it's sad. But so each of these is a story into of itself, but we're just going to do it on one slide. So we have multiple possible solutions to this idea of density and getting rid of polymers. The first is what we call the macropolymer approach, and rather than linking insulin to this insoluble polymer, we create a polymer out of insulin by linking individual insulin monomers to themselves with small light cleave linkers. And the result, and this image shows kind of a graphical representation of that. The result is a material where the vast majority is insulin, and the light cleavability is allowing that to degrade and release the insulin. And that material is about 85% dry weight insulin, so it's a huge improvement. A second approach we took is what we call the charge tag, and this is it's kind of a cool idea. We essentially shifted the isoelectric point of insulin from 5 to 7 by adding cationic groups that are linked through a light cleave linker. What does that accomplish? What it accomplishes is it allows you to formulate it under acidic conditions where it's totally soluble, but once it is injected into the skin at pH 7, it precipitates out at its isoelectric point. Then when you irradiate it, you release native soluble insulin. So these are some examples of these cationic additions that we added to insulin that conferred this change in isoelectric point. And I realize this is kind of getting into the weeds, but that's the whole conceptual basis for this particular approach. Maybe the most important thing about this is if you look at those materials, there are tiny numbers of atoms that have been added to the insulin surface that then accomplish the insolubility. And that's the key point. We're trying to make things that are very efficient, that are essentially almost all insulin with these very small additions that confer that light switchable solubility. And the final thing is maybe conceptually more straightforward. We link to insulin very nonpolar peptides or small molecules that just cause the whole final species to not be soluble. Now you can ask the question, well then how do you actually use it? Well, we can make these into particles. We mill them into particles that are injectable but are insoluble. And then upon irradiation, that little nonpolar hydrophobic tag is cleaved from the insulin, releasing native soluble insulin, which can be absorbed. So all of these materials now, we have materials that are able to release or able to contain about 90% insulin, give or take, or even more, whereas our first generation was about 5% insulin. So it's a huge increase in density. And then one more round of advancements. We've done a lot of work with increasing wavelengths of light. Everything up to this point has used a very short wavelength, deprotecting photocleavable group, and we're slowly working our way along the spectrum. We're now in the red, there's major advantages to that. Lower toxicity from the light, and secondly, greater ease of the light getting into the skin to access deeper layers of the skin and just have photolysis be more efficient. We've applied this whole approach to other proteins, to glucagon, which is the partner of insulin. It's sort of the push and pull of glucose homeostasis. And that's worked, and we've also found that in so doing, we actually address some of the problems of glucagon, including fibril formation. That's another talk. And then finally, recently, we found that we can get variable and multi-day response from one of these photoactivated depots. And in so doing, demonstrate that, which is a long-term aim, is that this can be used for multiple days. So I thought I would sort of end with a comparison of the two approaches, the pump approach versus the light source approach, and just do a side-by-side comparison. Pumps are much larger and are more unwieldy than a light source. They're about 20 times more voluminous. Light sources can be made very small, discrete. They're also much lighter, about 10% of the weight of a pump. Pumps are complex, they're mechanical. Light sources have no moving parts, and because of that, they're much less vulnerable to physical damage. They're much more robust. Pumps, again, this is the original motivation for that. Pumps are highly invasive because you're continually breaching the skin. Light sources have low or really no invasiveness. It's just light crossing the skin. The possibility, because of the nature of the cannula with the pump, you have the possibility of occlusion and infection, which is really not possible. And then this interesting observation that we had, which is that the delivery of or the release of insulin using the photoactivated depot is ultra fast, which we didn't anticipate, but it turns out to maybe be the most important advantage this approach has over every other advantage. And then there's the device cost. Pumps cost about $5,000. Now, part of that is probably a corporate decision, but they do contain a lot of moving parts and a lot of engineering, whereas a light source, I believe, can be made for much less money, about $20. It's all solid state. And if one is thinking about democratizing access to artificial pancreas, it's going to be hard to do at $5,000 per pump. Can we make a light source for $20? Well, sort of encouraged by, you can buy a pulse oximeter for about $20 on Amazon, and a pulse oximeter has a power supply, it's got an LED, it's got a detector, it's got circuitry, it's got a display, and they can make it at profit for $20. So all the essential elements are there, and there's a reason why that is. It's solid state. Once you figure out how to put that on a chip, it's very easy to do and very cheap to do. Okay, so then kind of bringing it all home here, to summarize, I hope I've convinced you that light is really a pretty powerful tool to control biological processes, because once you've linked a process to light, it's very easy to aim the light, to determine when the light goes off, and to determine how much light is applied to a given situation. Macromolecules, even though they're complex, are still molecules, and they're amenable to chemistry. You just have to kind of work through some of the details to figure out good, clean reactions that will allow it to make materials with the desired properties. mass spectrometry is an absolutely critical technology for this kind of work and, in biology in general, I would say. And then finally, in the specific application of the photoactivated depot, there are real, I feel, therapeutic, operational, and cost advantages to that approach. So with that, I'd like to just acknowledge all this work was driven by students in my lab. For the individual papers that were first author papers that I showed, I put asterisks next to the names, but everybody on this list is part of the group that has driven this forward. So we have to acknowledge all of their amazing work. We really benefited from Dr. Bill Gutile, who 20 years ago had the idea to bring high-end mass spec into our small department, and it's transformed what we do in our group and multiple groups have done. And that's been majorly supported by our administration, Dean Melchert, Professor Wyckoff. So thank you for that. My animal collaborator, Karen Cover, has been amazing. Funding from the NIH after a little hiccup at the beginning, and then early funding from University of Missouri and from the Dean's Fund. And with that, thank you for your attention. Thank you again for the invitation. And my understanding is we're going to break camp and reset up for a question and answer in a few minutes. So, thank you.
So before we transition to the question and answer part of the seminar, I really wanted to express our gratitude for sharing your research. And it's amazing to see the sophisticated chemistry interfacing with biology and hopefully in the future impacting patients with diabetes. So this is just a token of our appreciation. So this was fabricated from materials from within the state of Missouri and just represents what we're trying to do, impact patients here in Missouri and beyond. So thank you so much for your talk. I really
thank you very much. Appreciate that.
Yeah,
that's very, lovely. Yes. Excellent, excellent.
And there's a stand, and I'll move that later.
Okay.
So Dr. Emerson's going to be leading some questions, I think, in the Mentimeter, but if we have questions in person here or also in Columbia, so maybe we can kick it off with a Mentimeter question?
Yes, okay, so we have, how deep can a light travel? And are there implications for the particles or points of different sizes?
Yes, so that's a great question, and fortunately I have an answer for you. The original materials that we used, that use this ortho-nitro group, those are very short wavelength. They're in the far UV, so 360 nanometers, and the light does not, gets maybe a millimeter at best. So everything was done in the dermal layer. But I kind of referred to that earlier, that we're kind of working our way along the spectrum because we realized early on we really needed longer wavelengths of light because we really want to get into the subcutaneous because that's much more voluminous. Dermal layer is uncomfortable. You can, but if you've ever gotten certain vaccines, they go into the dermal layer, because a lot of connective tissue, it's painful. It also doesn't have much volume. So the subcutaneous layer would be much better. But to do that, you need longer wavelengths of light. But if you look at the literature, there's real indications that as you get to these longer wavelengths of light, you can get a huge increase in throughput. And so we've been working towards that. In fact, Michael Ansung, my student here, has a material that uses 500 nanometer light, and we've showed that it works in the subcutaneous for the first time. So that's definitely, that question is kind of pointing at an issue, but we're on it, and I think we're solving it. In fact, we're even going further along to 650 nanometers. Pretty soon we'll have, and that allows two things. A, we can access these deeper layers, which are the subcutaneous that's where normal insulin is injected, and it's much more voluminous, so it increases the size of the depot that you can have as well. So,
should we take one from in the room now?
Anybody in the room?
It's very, very excellent presentation.
Thank you.
However, one of my concerns is the functionality of the protein that is administered, that is insulin.
Yes.
Is it still biologically functional after the administration? Can you measure it? Can you withdraw blood from the animal, in this case animal, to actually test that it's still functional?
Sure. Well, in those original in vivo experiments, we looked at insulin presence in the blood, But then we also look at blood glucose reduction. And the only way you get blood glucose reduction is if it's retained bioactivity. And so we get full bioactivity. In fact, what we do is we compare, one of the first experiments we did was to take photolysed insulin, just that was in vitro photolysed, and inject it into the animal and to diabetic animals, and we demonstrated that it had full bioactivity. So that was a place where you're absolutely right. That could have easily gone wrong because we're doing, these reactions are done in some organic solvents, and things can easily go wrong. The one advantage is that insulin is a pretty robust molecule. It's got these 3 cross links. It's pretty compact, and that kind of helps us. It's maybe more like it's between a peptide and a protein, so that gives it some resistance to those problems. But yeah, we have demonstrated actual bioactivity. So.
The other question, may I?
No, go ahead.
The other question is, with regards to, obviously insulin has a receptor,
Yeah.
In your body. Have you tested your insulin on receptor that is in vitro? And to make sure that it's actually binding to the actual, to the correct receptor?
No, I mean, the counter argument is that even if we showed that, we would still have to do an in vivo demonstration. And we've sort of leapfrogged over that and just went straight for the in vivo because it could bind to the receptor, but for some other reasons not actually work in vivo. But my feeling is if it works in vivo, it must be binding to the receptor. And in fact, a secondary level of demonstration is that we detect this using an ELISA assay, a sandwich assay. It's going to be very hard for those 2 antibodies to recognize it if it's been scrambled in some way. It's still recognized by those 2 antibodies. So, but the in vivo readout to me is the most convincing thing. So, yeah.
So kind of on the receptors, can the receptors be susceptible to variable intensities of light? I don't know if it would matter at that.
I guess I need a clarification what is meant by that.
Yeah, all I got is.
I guess let me guess. There is a danger of phototoxicity. Even non-toxic light in sufficient quantities will cause things to catch on fire. So there is a dose limitation that we've kind of figured out what it is. So there is a danger of damage just due to light if you have a sufficient amount of light. So we know what that line is, and we're always on the right side or the correct side of that line. And that's part of the reason for going for longer and longer wavelengths of light, because that means you can use much lower intensities, because the light that you do apply, a much larger proportion gets through the skin and is then able to stimulate the materials. So.
Well, that was interesting, so we'll call it.
Okay, we'll call that a win. Okay.
Anything else in the room?
Yes,
Please.
so have you thought about or taken steps to combine this with the continuous glucose monitor? In other words, how do you get the readouts from the body's need and then?
Right. I mean, that is absolutely. I mean, that's the long term. We're seeing this as that's a long-term aim, is to integrate this into artificial pancreas, and we're seeing this as like a pump replacement, and ultimately we'd have to integrate with CGM people and algorithm people, and there are people who integrate everything together. They usually use existing pumps, existing algorithms, and then they do big clinical trials where they stitch all those pieces together. So we'd have to do some kind of approach like that. The way I conceive of this is we are trying to make the best approximation of a pump that doesn't have the problems of a pump, and then ultimately integrate it. But we haven't done it ourselves, although I'm sort of tempted in a way.
Well, I assume maybe the CGM could make a noise if you need some, and then you could personally activate the light for some amount of time.
Yeah, I mean, in fact, that's some of the artificial pancreas works like that, where it's sort of semi-automatic. It kind of gives alerts. You're going hypoglycemic or you're going hyperglycemic. And so there are CGMs that work like that give signals, and I guess it's sort of like full self-driving Teslas versus partially self-driving. You know, do you ever let go of the steering wheel? Yeah, but that is the long-term goal, is to kind of integrate into that sort of thing. But you know, like the point I made earlier in the talk is that all those pieces, there's been a lot of innovation. CGMs, you know, there are multiple companies. There are multiple academic groups. There are people really getting close to doing completely non-invasive. So I don't want to lock into something necessarily until, like, if those pieces get fully optimized, and then algorithm development is really, I mean, there are, you know, pure mathematicians who are working on this to really figure out very efficient systems for doing that. So I'm kind of sort of seeing what's winning, and then I feel like we're winning the pump domain, and then we'll try to integrate with.
Put a chip in the magnet or something like that.
Yeah.
That talks to the other thing.
Oh, that's in fact. So there are actually commercially available artificial pancreas systems, and they're a CGM and a pump and like an iPhone in between, doing Bluetooth between the two. And they do have clinical benefits, more time in range and so forth. But I mean, this is the interesting thing about glucagon, which I kind of just touched on, that it's this partner signal that increases blood glucose. And in fact, there are a lot of there not a lot, but people have pursued dual hormone artificial pancreases, so where you've got this push-pull, and then you can get a real titration of your blood glucose. But then you got two pumps and so forth. I mean, we had this idea of having one material with two different wavelengths, you know, one getting glucagon. It's a real challenge, and it might not be worth the fuss. But we have made a light-activated glucagon, and as I kind of referred to it, it's better than regular glucagon in the sense that it doesn't form fibrils, which regular glucagon, as soon as you mix it up, within a few hours it's inactive. So you have to mix it up, or you make a DMSO solution and you pump that into yourself, which is, you know, some concerns about that. But our photoactivated depot of glucagon resists fibrillation because I think the attachment kind of physically prevents the individual monomers, and yeah.
Great.
So,
You go ahead.
this is a very simple question. With light, what are the ambulatory effect? Can you move around?
Yeah.
You know,
That
administration,
ambient light, yeah. So that's a concern. There are a couple ways of dealing with that. One is just having your light source on top of the injection site, shadowing it so that no ambient light hits it. And you'd want the light source on top of it anyway to provide the light that you need. So that you can do that maybe like a watch band or something, or maybe a small amount of like an adhesive patch so it just sticks there. The other aspect of it is to do with fine-tuning of the photochemistry. Our light sources can get, there's a narrow band of wavelengths that we need, and we can kind of put our energy in those, and the light source can deliver a lot of light in that narrow range, more than even ambient light. I mean, the sun is more powerful, but that covers the entire spectrum and beyond, you know, because UV as well as infrared. So that has a lot of energy in it, but in terms of the narrow band that we need, we can actually have higher intensities in our light source. And we actually looked at that. We took spectral readings and compared it with what our light source can put out. So we can actually fine-tune some of that by fine-tuning the photochemistry, essentially.
Time for one more?
I'm fine. I can go all day.
Let's do one more.
Have you considered controlling gene expression with this approach, using light to treat a different specific medical problem? What disorders might be suitable to this approach?
Ooh, wow. Maybe they can tell me. I mean, I always conceived of that as very much a biological tool that for picking apart biology, and but in terms of disease, although, you know, you have to be careful about saying it can't be used in that way, because then you have to just think harder potentially. So there could be disease states. I haven't really pondered or thought of any in particular. I was really aimed at something that would allow biologists to really spatially and temporally and manipulate gene expression to understand the role of different genes in processes. Yeah, so, but it wasn't really aimed at disease. So.
Great. Well, thank you so much for your talk.
Yeah, thank you all for coming.
And just for everybody's awareness, we do this every month. Next month is May 14th, and we have Dr. David Fleming, who's going to be talking about complex decision-making at the nexus between precision medicine and end-of-life care. So should be very provocative and maybe a little bit tough topic to listen to. So I'm very excited about that. So thank you so much.
What's gonna be the venue?
So next month it is in Columbia. Yeah, so we roam around, so.
But it's on Zoom. It'll be.
But it's on Zoom,
Yeah.
yeah, so you can always join. Thank you so much for coming.
Great, thank you, thanks, and thanks for the invitation.
Yeah.